All right, hello and welcome. The purpose of this video is to discuss the Crossbridge cycle. Of course, in order to understand the Crossbridge cycle, we need to define the functional unit of the muscle itself, which we previously talked about. This is the sarcomere. And the sarcomere consists of, if we were to label a few things, of course, the boundaries are Z discs. Attached to the Z disc, we have the thin filament, shown in blue, and over here in red, the thick filament. And then anchoring the thick filament onto the Z disc is shown here in green, a protein called titin. Muscle contraction is no more and no less than the thick filament grabbing onto the thin filament. We'll show one of these. It'll reach up and grab onto it, and then it will pull in that direction. Of course, at the same time, we're grabbing on this side and pulling in that direction. So the muscle contraction itself occurs as the width of the sarcomere decreases by up to 50%. This Z disc is going to move to the midline. This Z disc is going to move to the midline, and the whole thing compresses. That's muscle contraction. So to understand Crossbridge cycle, what we need to understand is what's going on right here. This interaction between the myosin head, which is the thick filament, which is part of the thick filament, and then the thin filament. See what's going on. Now, previously we've discussed this. If we were to zoom in on this thin filament, let's see what we might see. The thin filament itself is composed of two interwoven strands. We see these here of F actin. There are two strands. They're woven in a helix. And if we were to zoom in on one of these strands, this is what it's going to look like. The F actin itself is composed of individual G or globular G actin monomers. And we've talked about this already. Here in yellow, we see tropomyosin. And the role of tropomyosin is to hide or mask those binding sites on the individual G-actin monomers to which our myosin head could bind. And lastly, the important player, troponin, which is calcium sensitive. And when calcium comes in, calcium binds to troponin, forces a conformational shape change, a shift. And because troponin is bound to the F-actin and also the tropomyosin, this shift in shape pulls tropomyosin off of those binding sites and exposes them so that myosin can bind. So, in comes calcium as the main player, and we talked about in the neuromuscular junction, the end game is to have calcium released from the sarcoplasmic reticulum. Calcium comes in, binds to troponin. This has the effect of moving tropomyosin out of the way. Once tropomyosin is moved out of the way, these binding sites on the individual G-actin monomers are exposed. And that allows myosin to go in and bind to the closest one. This is where we start the Crossbridge cycle. Now, before we continue, one thing that I want to do is just make sure we understand a little bit about myosin. Myosin is a lot like a mousetrap. If you've never played around with a mousetrap before, it's this orange bar right here. That's the deadly portion. And this spring will pull on that orange bar because the orange bar itself would rather be on this side. In other words, that orange bar in a resting state is over here. But to set it, what do? We reverse that. We pull that orange bar over to this side, loading potential energy into that spring. And then we use this clasp, shown in yellow, to fold over the orange bar, and we lock that clasp into place. We then set a piece of delicious cheese on top of that lock, disguising the trap. As the mouse comes in, takes the cheese, that moves the lock out of the way, and the force, the potential energy stored in that spring, forces the clasp out of the way as that mouse trap quickly snaps closed. And if the mouse is lucky, the mouse got out of the way. Myosin works in much the same way. As the picture depicts, myosin exists essentially in two different phases. These are not official terms, but to continue the analogy with the mousetrap, this is in the set phase where there's a lot of potential energy stored in that myosin head. And this is in the sprung phase where that potential energy was released and that myosin head snaps forward. Now that we understand the basics of how myosin works and its ability to bind to F-actin, let's now explore the Crossbridge cycle. The Crossbridge cycle can be broken down into 4 steps. The first is called Crossbridge formation. The second is called the power stroke. The third is a step called release. And the fourth is what we call recovery. Now let's go through these one by one. So, step 1, Crossbridge formation. A couple of things to note. Number 1, this happens in the presence of calcium. Why? As we just reviewed, calcium binds to troponin. Troponin then moves tropomyosin out of the way, exposing those binding sites on F-actin. So, the crossbridge formation occurs as myosin binds to actin. All right, what does this look like? Well, as we show myosin, there's a couple of things that I want to bring to your attention. The presence in the myosin head of 2 different molecules. Here shown in blue, this is ADP, or adenosine diphosphate, and shown in white, this is another phosphate. Because it's not bound to any carbon-based molecule, we call it inorganic phosphate, PI. PI is actually the locking mechanism that is holding this myosin head in a set state. Now, as we said before, with tropomyosin moved out of the way, each one of these G-actins has a binding site on it for myosin. This is crossbridge formation, when myosin binds to one of these binding sites. The interaction between the myosin head and the G-actin binding site is just like the mouse chewing on the cheese. This is what disrupts the lock. So once we have the interaction in place, that's going to lead to step number 2, the power stroke. The power stroke is triggered by the release of our lock, which is inorganic phosphate. So returning to our picture, we've got crossbridge formation. It's that interaction that causes the release of our inorganic phosphate. With inorganic phosphate released, that was the lock holding it in place. What we see is a quick ratcheting of the myosin head to its native state. It snaps just like a mousetrap. In the process, we also lose the ADP. And of course, because this is bound to the to the actin thin filament, this whole filament is displaced in that direction. At the end of the power stroke, recognize the total displacement toward the midline, and also recognize that the myosin head is no longer bound to inorganic phosphate, nor is it bound to ADP. Also, please note, this did not require ATP energy. Okay, the energy for this was the spring-loaded energy, the potential energy stored in the myosin head. This is a common mistake that students make. No ATP required. And forgot to write this down, the release of ADP. That's the power stroke. That's a muscle contraction. The last two steps, release and recovery, just have everything to do with resetting the process so that I can do it all over again. For step 3, release, this is where ATP comes in. So this step requires ATP. If we go back and we look at our picture, what does this look like? ATP comes in, and ATP is going to occupy the site that was vacated by ADP when ADP was released during the power stroke. So in comes ATP. We'll bind it, but ATP is a little bit bigger and a lot more negative than ADP, and this causes the myosin head to change its shape slightly. Now it doesn't like binding to actin. And so this has the entire effect of letting go. We can see now that in the presence of ATP, there's no interaction between the myosin and the actin. This is release. Of course, this is what we see with rigor mortis, where the body's no longer producing ATP, and in the absence of ATP, myosin can't let go of actin. This is what the stiffening of the muscles is associated with in that roadkill that you saw the other day. All right, now to finish things off, the last step is recovery. In other words, we've got to reset the mousetrap. And this is all about ATP hydrolysis. As we explore this, maybe it's just a good time to review what ATP is all about. So if we drew ATP real quick, adenosine, it's a pentose sugar attached to adenine, and then out here on the 5' carbon, 1, 2, 3 phosphates. Phosphates are negatively charged. So this one has a big negative on it. This one has a big negative on it. To overcome, right, opposite charges attract, but like charges repel. And so in order to form this bond, I have to make 2 negative charges essentially hold hands. They're not really happy about it. But then I got this third phosphate, and he's got a couple of negatives on him, and I'm asking this phosphate to hold hands with this phosphate. There is a lot of negative charge up there, and these phosphates don't like being that close together. It takes a lot of energy to put them together. This is what metabolism is all about, making these bonds, but they're not happy. So because making bonds requires energy, specifically making this bond requires energy. So hopefully it makes sense, at least at a basic level, that breaking that bond releases energy. And we can draw this out as a chemical equation where we have ATP being broken down into ADP plus the phosphate that was broken off, inorganic phosphate, plus energy. And it's this energy that we're going to use to reset the trap. So here's ATP. The enzymatic activity for this reaction is actually within the myosin head itself. So the myosin head is going to break that ATP so that now, instead of ATP, we've got ADP, and we have our inorganic phosphate. And the energy that's released by breaking that bond is what resets this head And we can now move it back to the cocked position, where inside we've got our ADP and our inorganic phosphate. And now what I hope you can recognize, if we zoom out, we are right back where we started from, ready for another round. And if there's if there's an actin binding site available because calcium is still present in the cytoplasm, then it's going to bind again and it's going to ratchet again. However, if calcium has been removed, allowing tropomyosin to recover those binding sites and mask them, this is how the myosin head stays, ready to engage as soon as a binding site becomes available.